it's really amazing that we see specifically this area, this third ventricle area in the hypothalamus, because it's a very unique part of the brain that performs some pretty special functions. And what we were able to detect was just thousands of different genes changing with age. It's actually one of the few places in the brain that has a leaky blood-brain barrier.
Everyone, and welcome back to Teach Me in 10. Today we have Dr. Kelly Jin from the Allen Institute, and she's going to tell us all about age-related changes in the brain at the cellular level. So can you explain why understanding how the brain changes with age is so important, and how do we actually study it?
So aging is the greatest risk factor for all of the major chronic diseases that are currently prevalent in the developed world, including diseases like cardiovascular disease, cancer, type 2 diabetes, neurodegenerative diseases. The single greatest risk factor for getting all of these diseases is getting older. And so the idea is, if we can better understand not just what's going on inside of our bodies when things go wrong and result in these diseases, if we can better understand just what happens as we age, just during the normal, healthy aging process, perhaps we can better not only understand and be able to better treat these diseases, but ideally even prevent the onset of all of these diseases simultaneously.
So, what actually are some of the most noticeable effects of aging on the brain?
Firstly, we took a single cell approach to look at aging. So we took the brain and broke it down into its individual component parts, in this case, individual cells. And one of the strongest reasons for doing that is that the brain is actually extremely heterogeneous. So it's made up of a lot of different kinds of cells. And so it's important for us to be able to break down the brain into those component parts to be able to tease apart how these different cell types are changing with age. And what we were able to detect was just thousands of different genes changing with age. And interestingly, the majority of these genes are uniquely changing in one or two cell types. And so what that kind of means is all of these different cell types are changing with age in different ways, which kind of further supports this approach to looking at aging from this high resolution single cell perspective. Across those different cell types, at least the mouse brain is actually made up of mostly non-neuronal cell types. The number of non-neuronal cells greatly outnumber the number of neurons in the mouse brain. So by non-neuronal cells, this includes cells like oligodendrocytes, astrocytes, which are widely regarded as kind of these support cells for neurons, as well as microglia, which are the largest immune cell population in the brain, as well as the cells that make up the vasculature and the cells that make up the ventricular system. And interestingly, these non-neuronal cells actually showed larger numbers of gene expression changes than most of the neuron population. So that tells us that we can pay kind of more attention to some of these non-neuronal cell types, at least in the context of aging.
How do these specific cell types in the brain undergo this significant gene expression changes as we age then?
The data set is so rich, and there's even the paper that we just released kind of just scratches the surface at the different kinds of changes going on. But I can go through some of my favorite stories. So we saw increases in gene expression related to inflammation across a lot of those non-neuronal cell types, especially the microglia, the immune cells. And we saw increase in not only inflammation genes, but also genes related to immune response and cell motility, suggesting that these immune cells are becoming more active as these mice get older. We also saw generally across neurons and some of these non-neuronal support cells like astrocytes and oligodendrocytes, decreases in gene expression of genes related to neuronal signaling and neuronal function and structure, suggesting that there's kind of a general gradual loss of neuronal signaling integrity across the brain. There are also small populations of what are called immature neurons throughout the brain. So these are small neuronal populations that continue to create new neurons even into adulthood. And interestingly, a few of these populations seemed to be depleted in the older mice, so we weren't able to see them at all, suggesting that the ability of these mice to make new neurons even into adulthood seems to get worse with age. And interestingly, we saw a lot of highly specific, transcriptionally specific cell types around a very specific region in the brain called the third ventricle, which is located in a very critical part of the brain called the hypothalamus. A lot of cell types around that region also showed lots of gene expression changes with age. And interestingly, a lot of those cell types are very well known for the role they play in sensing nutrients and energy homeostasis and controlling feeding behavior. Cells like tanocytes and ependymal cells, also neurons that express receptors for very famous hunger hormones like leptin and GLP-1. So that was a really interesting observation to see all of those cell types around that same region kind of show a lot of these changes with age.
All of what you just said, you mentioned in your recent paper about there being a concept of certain areas in the brain being potential aging hotspots. Are these the kind of things that you mean? Is that what aging hotspots means? And how are these changes in these areas are associated with aging?
Yes, that is essentially what it means. So observing kind of these areas in the brain that we didn't expect to have a lot of very specific cell types showing greater gene expression changes with age. And it's really amazing that we see specifically this area, this third ventricle area in the hypothalamus, because it's a very unique part of the brain that performs some pretty special functions. So, as many people know, most of the brain has a very, very tightly regulated vasculature system, meaning that the blood vessels, in most of the brain, the blood vessels are very tightly regulated and they don't allow many things in the blood to penetrate into the rest of the brain. And that's mostly to protect the brain tissue from any kind of pathogens and things that are in the blood. This is known as the blood-brain barrier. This region in the hypothalamus that I was just talking about, it's actually one of the few places in the brain that has a leaky blood-brain barrier. So that is to say the blood vessels in that region of the brain they have more gaps. So they allow more hormones and metabolites that are flowing around in the blood to penetrate into that part of the hypothalamus, thereby interacting with different neurons, which kind of allows the brain to sense what is going on in the rest of the body, kind of sensing these molecules that are trying to communicate how the brain should be responding to whatever is in the environment. And so the fact that we see a lot of changes in these cell types that play a really critical role in that kind of sensing is really amazing and kind of points us to hypotheses about this particular process, the brain's ability to kind of integrate signals and communicate with the rest of the body might be one of the first processes to start to decline as we age.
Earlier you were talking about leptin and food behaviors, and in your paper there's a lot of mentions of potential links between diet, metabolism and brain aging. So can you share what your research suggests about these connections?
So there's an enormous body of literature out there about the relationship between diet and aging and lifespan. There's a lot of very amazing studies that have shown that essentially if you feed something less, not all the way down to the level of malnutrition, but just generally feeding something less tends to make things live longer and also appear healthier. And this has been demonstrated in just about every single model organism that's been tested in the lab. And so there's a huge amount of academic and therapeutic and also commercial interest now in trying to understand how that relationship works, how that really tightly regulated relationship between diet, metabolism, and aging and longevity, like what exactly is going on. And so I think our study is really, really exciting because we took this pretty unbiased approach, pulling down as many different kinds of cells as we could from the mouse brain and asking, okay, across all of these different cells, which ones are showing greater numbers of gene expression changes with age, and how are they, different ways in which they're changing. And even with that unbiased approach, it kind of still pointed us to these cell types, which are famous now for their role in controlling our feeding behavior and how hungry we are, and energy homeostasis. And so I think our study is really exciting in that it kind of reveals this piece of the puzzle, this important piece of the puzzle, which is how the brain really plays a role in this tightly regulated relationship between diet and longevity.
Amazing. Thank you so much for joining us today, Dr. Jin. I'm really excited to see what your lab does next.
Thank you. Thank you for the opportunity to share our work.
A big thank you to Dr. Jin for taking part in Teach Me in 10 today, and a big thank you for all of you watching. Until next time.